This is an arterial blood gas printout from a crashing patient. The values highlighted in red indicate a severe physiological crisis, a pH of 7.15, a bicarbonate level of 12, and a PaCO2 of 30. These numbers define a metabolic acidosis, a pathological state where the accumulation of fixed acid depletes the body's bicarbonate reserves and drives down the arterial pH. At the microscopic level, rising hydrogen ion concentrations disrupt the precise electrostatic bonds that maintain protein structures. Critical enzymes across every organ system begin to unfold and denature. Macroscopically, this manifests as depressed myocardial contractility and extreme arrhythmogenicity. The heart muscle rapidly loses its coordinated pumping rhythm, falling into a chaotic, non-functional spasm. Survival hinges on preventing this lethal protein denaturation during a massive acid influx, buying time before the kidneys can eventually clear the load. The body's response to this threat is a highly choreographed, time-dependent cascade. This timeline is the fundamental framework for understanding acid-base defense. It is divided into 3 distinct phases: seconds, minutes, and days. The first phase is immediate chemical buffering, occurring in seconds. This is followed by intermediate respiratory compensation, which activates over minutes to hours. The final phase is delayed but definitive renal correction, which takes days to complete. Mastering this chronological structure is necessary for diagnosing mixed acid-base disorders and predicting when a patient is about to decompensate. The primary extracellular buffer in this first line of defense is the bicarbonate system. This titration curve reveals a biochemical paradox. According to the Henderson-Hasselbalch equation, bicarbonate has a pKa of 6.1. At a physiological pH of 7.4, the system operates on the steepest, lowest capacity part of the curve, far from its point of maximum efficiency. In a closed chemical beaker, this system would rapidly fail. It would reach equilibrium and stall as the bicarbonate was consumed. Human physiology overcomes this by operating as an open system. The lungs act as a mechanical bellows, continually venting carbon dioxide gas out of the system. Simultaneously, the kidneys independently control the bicarbonate base. By regulating both the acid and base components through separate organs, the body prevents chemical stall and maintains pH balance despite the system's inherent mathematical limitations. Bicarbonate's dominance as a buffer relies entirely on this dynamic organ excretion. As extracellular bicarbonate is consumed, excess hydrogen ions move toward intracellular protein and phosphate buffers. This creates a problem for electrical neutrality. As positively charged hydrogen ions force their way into the cell, they push positively charged potassium ions out into the bloodstream. This mandatory exchange is required to maintain transcellular electroneutrality. This cellular shift explains why acute metabolic acidosis typically presents with clinical hyperkalemia. The body sacrifices stable potassium levels to temporarily halt lethal intracellular acidification. Hemoglobin is the primary non-bicarbonate buffer of the blood. The hemoglobin molecule contains 38 histidine residues. Each residue features an imidol ring that can bind a free hydrogen ion. With a pKa of 6.8, these imidazole rings are highly effective buffers at the pH levels typically found inside red blood cells. Under the Haldane effect, hemoglobin's buffering capacity changes based on its oxygenation state. Deoxyhemoglobin, with a pKa of 8.2, is a better proton acceptor than oxyhemoglobin. When oxygen detaches, the protein shifts shape, allowing the imidazole ring to more easily capture a hydrogen ion. This creates a physiological synergy. As hemoglobin delivers oxygen to acid-producing tissues, it simultaneously becomes optimized to sponge up the resulting acid waste. Chemical buffers buy only a few minutes. The respiratory system must provide the next defense phase. Falling arterial pH is detected by peripheral chemoreceptors in the carotid and aortic bodies. These sensors send immediate signals through the glossopharyngeal and vagus nerves to the medullary respiratory center. Central chemoreceptors in the medulla are initially blind to metabolic acidosis because the blood-brain barrier is impermeable to hydrogen ions. even as carbon dioxide crosses it freely. The medullary center triggers an effector response, extreme alveolar hyperventilation, clinically recognized as Kussmaul respirations. This aggressive breathing clears volatile carbon dioxide to restore the ratio between bicarbonate and arterial CO2. On a Davenport diagram, the initial acidosis moves the patient to the left. The subsequent hyperventilation slides the point downward across the CO2 isobars, pulling the pH back toward the normal range. This defense is limited by the work of breathing and central hypoxia, which generally prevent the PaCO2 from dropping below 10 to 15 millimeters mercury. Respiratory compensation is a powerful bridge, but it is an adjustment of ratios rather than a permanent clearance of acid. Permanent correction requires the kidneys to physically excrete the fixed acid load. The kidneys must first reclaim 100% of the bicarbonate filtered at the glomerulus. In the proximal tubule, a sodium-hydrogen antiporter pushes hydrogen into the lumen to initiate this reabsorption. This hydrogen combines with filtered bicarbonate to form water and CO2, which diffuse into the cell to reform bicarbonate. This bicarbonate is then returned to the bloodstream. Once the filtered bicarbonate is saved, the kidneys turn to the physical excretion of the remaining fixed acid load. The kidney traps excess hydrogen in the urine using phosphate and ammonia buffers. These hydrogen ions are bound and permanently flushed from the body. For every hydrogen ion excreted this way, a new bicarbonate molecule is generated and added to the blood. By manufacturing this new buffer base, the kidneys permanently cure the acidosis, a definitive correction that takes days to complete. Viewing the physiological cascade from a macro perspective reveals an integrated defense sequence. The immediate phase relies on chemical buffers acting as shock absorbers. The intermediate phase uses hyperventilation to adjust the acid-base ratio. The final phase provides definitive cure through renal excretion and buffer regeneration. Board examiners look for this specific chronological hierarchy when evaluating a passing answer. This chronological framework allows you to track the progression of a crashing patient and anticipate their physiological exhaustion before it happens.